Hi, my name is Ollie, and I have the pleasure of introducing to you today our new search engine extension called Kyper that we launched with Spectronaut 20 that is integrated in our directDIA search pipeline. So what is Kyper and what is it good for? So a couple of months ago, we were looking at the problem of unspecific search spaces, and especially on the way how unspecific search spaces grow in their complexity with increasing peptide length that you're adding to your search space. So if you look at a small search space of only seven peptides, you can see that you're already at about 178 times more peptides in your search space compared to a cryptic search space. This grows all the way up to almost 900 times larger search space compared to a cryptic search space if you go all the way till length 50, 52 for your peptide length distribution. Obviously, this increase in search space also leads to a massive increase in search time. So you can see here that for an example cryptic search, you usually are done within 10 minutes in terms of search time, while the same sample searched with an unspecific search setting would take up to 64 times more time. In this case, about 420 minutes in total. So the thing we came up with for improving this is what we call Kyper. And it's our new search engine that was built from the ground up, basically, as an unspecific search engine. So we basically took the idea of a novel MS2 indexing concept and thought, how would a search engine look like that is purpose-built on the idea of unspecific search spaces, basically. And what we came up with is what I already mentioned, we called Kyper, and it's integrated in directDIA in a way that it's basically working together with Pulsar. So every time Pulsar gets MS2 scans to search, it will send them to Kyper, which will do a very fast MS2 index-based analysis of the scan to return a list of suggested peptides it saw in the scan. These peptides are then fully enumerated in Pulsar again and scored, usually using our usual scoring methods. The whole thing is quite fast and has a very low memory footprint, which is very nice. And it also benefits from some other new scores that we added specifically for the subset of immunopeptidomics data. So in terms of runtime improvements, they are quite staggering, in my opinion. So you can see here, for example, for a class 1 immunopeptidomics search space, 7 to 12 in length, you can see that we already reduce in roughly 70% in search time. And we're now able to do a full directDIA search, start to finish, including all steps like file pre-processing and post-processing steps. We can do this now in about 8 minutes per DIA file for immunopeptidomics class 1, which is a 70% reduction in search time. For class 2 immunopeptidomics, the drop is even more significant, where we can go all the way down to minus 87% in search time for class 2 immunopeptidomics, and also reach search times of roughly 10 minutes per raw file now for a directDIA search. This is quite nice, but obviously just being fast is not very helpful. Is the data also any good? And here I can proudly proclaim now that yes, it is. So we do also gain massively in terms of peptide IDs compared to Spectronaut 19. For class 1 immunopeptidomics, we can see up to, like on median, 75% more peptides identified with directDIA in Spectronaut 20 using Kyper. And for class 2 immunopeptidomics, it's still impressive, plus 18% more peptides using a directDIA search in Spectronaut. Obviously, an important question about when raising identifications is what the quality of these identifications are. And we went to great length to evaluate our false discovery rate. And here you can see an example of 2 different entrapment strategies that we're using, one being a 2-species test using human and Athaliana with an unspecific search, and the other one being a 10x entrapment strategy, a synthetic entrapment strategy. Each of the 2 data sets was run in 4 different filtering steps and 6 different bootstrap reanalysis, basically to get a feel for what the reproducibility of the FDR is. And all searches were run without any protein level FDR control, since this is not something that is commonly done for immunopeptidomics. As you can see here nicely, that pretty much all of our data sets and replicates that we measured were nicely below the expected 1% FDR control, sorry, not 1%, but the expected FDR controls for the different selected filter levels that we applied. One other really cool thing about Kyper is it's not only a fast unspecific search, but it's by design an open modification search engine, which means that you can now do an unspecific open search on, for example, in this case, a human cell lysate, which is an astronomically large search space. So you can see here that with Spectronaut 19, with a closed search, unspecific closed search, we ran the sample in about 440 minutes, while with Spectronaut 20 in the closed search, we're down to 28 minutes, while still gaining quite significantly in terms of identifications. But the real killer here is that with an unspecific open search, we are actually only gaining a very small amount in terms of search time. And considering that we have about 35 different common sample prep and biological modifications that we added to our search space. So the ability to do an unspecific open modification search is something that, in my opinion, really opens a lot of cool new experimental designs that you can now investigate. Furthermore, what is nice for the unspecific open search is you get, so this is basically how it looks like in the software. You can select the PTM probing search in Spectronaut and select which variable modifications you want to add. By default, as I said, we populated 35 common ones, but you can change this setting to whatever you want. And also what is really nice is that you get a nice distribution of what the most common modifications were that you observed in your sample. So to make an informed decision, should you follow up with a closed search. Additionally to our improvements in the general search pipeline, we also added a couple of new modifications, primarily towards investigating immunopeptidomics data. Like, for example, we have a motif analysis plot now in Spectronaut. Additionally, also length distribution plots, and also a very nice identification overlay for diapassive methods, to where you can see which identifications in which charge states you recovered from which region of the data. All in all, I think we have a really solid new workflow that we released now with Spectronaut 20, and I'm very excited to hear your feedback, essentially, on how it works in your hand. Thank you very much.
As you have seen, we have improved in identifications for immunopeptidomics in Spectronaut 20 quite a lot. So we wanted to validate whether the newly identified peptides are actually good peptides, biologically relevant peptides, and so on. So to start with, in immunopeptidomics, the peptides are actually produced within the cells by endogenous digestion of proteins by proteasome. So proteasome creates short peptides that are then loaded to MHC class 1 molecules, which are presented then on the surface of the cells. These peptides are then directly isolated and measured with mass spectrometry. Because of this, the immunopeptides have specific characteristics. They are unspecific peptides. They have specific motifs, which means that at specific positions, certain amino acids are overrepresented. This makes these peptides quite unique, but these motifs differ a lot between different alleles that the peptides bind to. Because of their characteristics, you can also use certain tools to predict whether the peptides would be a good or a bad binder for certain MHC class 1 molecules. The peptides have specific peptide length distributions with the enrichment mostly for peptide of length 9. We can also have various sample complexities ranging from monoallelic cell lines, where you would expect a single motif, to multiallelic cell lines and complex patient samples. Because of different alleles present in the population, we also have large population variability. So analyzing large biological patient cohort can be very challenging. Because of this, traditionally DDA was used to analyze immunopeptidomic samples. However, we wanted to see whether we can tackle the same questions with DIA data in a direct DIA mode. To test this, we collected a wide variety of publicly available in our internal samples, ranging in complexity from monoallelic to multi-allelic cell lines, plasma samples, and patient tumor samples. We analyzed several data sets, but I will only focus on a few key results today. We analyzed these samples in library-free mode, but we also compared it to the analysis that you get with library-based analysis if you create a project-specific DDA library first. We also looked specifically into the results if you only analyze DDA data directly. With this, we wanted to check whether we can, whether directDIA can identify good binders, whether we can recapitulate known motifs and peptide length distributions, whether significantly different samples can all be searched together, and whether we can measure the variability between different samples directly. To start with, we started with the simplest model you can have with monoallelic cell lines. We took this published dataset where C1R cells were transfected with either allele A or allele B. With this, they generated condition A and condition B samples that would only have allele A or B or low level of expressed allele C. We analyzed everything together in a single search in a library-free mode with Spectronaut 20. And with such analysis, you would expect the following results. So we used MHC flurry to predict which alleles the peptides would bind to. And in such analysis, you would expect that in condition A, you would only observe peptides that bind to allele A or allele C. In condition B, only peptides that bind to allele B or C. And for the overlapping peptides, they should only come from allele C. We looked at the results for Spectronaut 19 and 20, and we were very happy to see that while in Spectronaut 20, we almost double in identifications, we still get the expected picture. So we have a very small number of overlapping peptides, and those mostly come from the allele C. So we then calculated what is the percentage of unexpected peptides as a proxy for run level FDR. So we would, with applying 1% run level FDR filter for precursors, we would expect to not have more than 1% of unexpected precursors in a specific run. Looking at the averages across the runs, you can see that we indeed do not go above 1% with our approximation of run level FDR. And if we apply stricter PEP filters, you can be even stricter with this and you get even less unexpected precursors. However, since you are stricter with the PEP filter, as expected, we also lose some true positive identifications. So this looked very nice for the simple model of monoallelic cell lines. So we wanted to see whether we can see the same thing for a more complex sample, in a more complex sample. We looked at, again, published dataset of multi-allelic cell lines. In this case, A375 multi-allelic cells were used, and they were used in increasing amount of the starting material. So you have this concentration range. The peptides were isolated and measured in DDA or DIA mode. We had 9 DDA files available and 18 DIA files with 3 replicates of 6 concentrations. We then search everything again with Spectronaut 20 in library-based and library-free mode. Looking at the results, we can first see that across the concentration range, identifications indeed increase, which makes sense. But what was even nicer to see is that library-free analysis always yields more identifications than library-based search. However, if we look at the overlaps of the identifications, we can still see that we have very good overlaps and we identify more unique identifications in our library-free mode. What is more, we wanted to check whether the biology makes sense. So we looked at the motifs. So those are the 6 alleles that are present in this cell line. And these are the expected motifs that you would expect for these alleles. So on the x-axis, you can see the amino acid position, and on the y is the overrepresentation of specific amino acid. And looking at our results for library-based and library-free analysis, you can see that firstly, the two searches agree with each other quite a lot. So we get very similar motifs between the two searches, but we also see very similar results to what we would expect these alleles to look like. So we can even though we identify way more in directDIA, we can still recapitulate the same biology. We tested this as well with looking at the peptide length distributions. So what you can see is that we see very different peptide length distributions between different alleles, which is something that is also known in the literature. However, between the two searches, we do not see significant differences. The same is true for allele ratios. So we observe very similar allele ratios, no matter whether we do a library-based or library-free search. This shows that we do not introduce any significant biases in terms of peptide lengths or in terms of alleles with our directDIA. So we can recapitulate the same biology again. This dataset was interesting as well because we could also test quantification. So they use different concentrations of the starting material. So we could take the known concentrations and correlate them with the measured abundances. Fitting a linear model through the points, we could then calculate the R squared of the for each individual precursor. And here are just 2 example precursors. You can see, so that you can see how an R squared of 0.75, for example, looks like. And I think you can see nicely that we get very similar fits for the different searches. And in general, the fits look pretty good. We did this for all the precursors where at least 3 quantitative values, so at least 3 concentrations were quantified, and then displayed an overall picture in this distribution. As you can see, the distribution of R squared, the majority of the precursors actually has a very good fit. And what is more, we see that we get way more precursors with a good fit for the library-free analysis. So we don't only identify more precursors in general, but the precursors that we identify in directDIA are also well quantified. So this all looked really good. So we went one step further and wanted to see whether directDIA can also be applied to a complex patient sample study. With this is our internal data sets that is very fresh, but the data looked very exciting, so we wanted to show it to you. We had 11 patients where with small needle biopsy, the samples were taken either from the tumor, from the metastasis, or from the normal tissue. Samples were then measured on a timsTOF Ultra 2 with 20 samples per day method. Samples were measured in DDA and DIA. And then again, we analyze everything with Spectronaut 20 in library-based, library-free, and DDA search. The samples were also HLA typed. So you can see for all the patients, we had information which HLA alleles they had. And I hope you can appreciate that the samples, so the patients were very different between each other. We had a wide variety of different alleles that were all analyzed together. First, looking at the identifications, you can see on the x-axis, so here we have all the 11 patients, and the results we got from the DDA files, from library-based analysis and library-free analysis. We observed that we have various very different number of identifications between the different patients. However, we observed a similar trend for all the searches performed. Further, did library-based analysis always performed better than looking directly at the DIA files? However, library-free analysis outperformed mostly both library-based and DDA analysis. So we always got more identifications with directDIA. Even though we then looked as well at the fraction of good binders. So we can see for all the patients, for directDIA, we had more than 70% of peptides classified as good binders. And the fraction of good binders is very comparable between the different searches. So even though we get way more identifications, we still maintain a large fraction of good binders. Next, we looked again at the allele ratios and peptide length distributions. So here the samples were way more complex. So we can really nicely see that we observe a wide variability between the patients. So in allele ratios, you can see that some patients have more identifications from allele A, some have more identifications from allele B. And while the patients are very different, we come to very similar conclusions if we look at DDA or DIA files. So between the searches, we do not observe huge differences, but between the patients we do. The same is true for peptide length distributions. You can see that we can clearly observe differences in peptide length distributions between the patients, but focusing on a single patient, you can see that all the searches give us the same conclusions. Lastly, we looked at the overlap of identifications for our directDIA identification. As I said, all the patient samples were searched in a single analysis. So we wanted to see how many of the overlaps can we observe. Interestingly, you can see that the majority of the peptides were actually identified in a single patient, for example, here. And then we observe some overlaps between individual patients as well. So if we first focus for patient 11, you can see the motifs that we get from these unique peptides are like this. And then if we look at the HLA typing from patient 11, there are 3 alleles that are unique to this patient. And the motif A and B look like this. So you can see that if we combine those 2 motifs, we essentially get the motif plot of our unique peptides. So this is very cool to see, but we wanted to see whether we can get something from a more complex behavior. So for example, here where we have even overlap between 3 patients, those are patients 6, 7, and 11. You can see that they share some alleles, but there is a single allele where all 3 patients have it, but none of the other patients do. So if we look at the motif of overlapping peptides, I hope you can appreciate that it matches really well to the expected motif of the overlapping allele. If we move further for looking at the next one, where we have an overlap between patient 9 and 10. Again, this is the motif that we get for the overlapping peptides. And while these patients share several alleles, there is a single allele that is unique to those 2 patients, and this is the motif of this allele B. And again, you can see that the motif matches really well the overlapping peptides motif. And with this, I hope I could show you that essentially it's really cool that in a single search, we can analyze all these very different patient samples together and still get very meaningful biological results. And we do not get a lot of transfer of identifications between the patients. So with this, I hope I could show you that directDIA in Spectronaut 20 leads to increasing identifications of biologically relevant immunopeptides. I showed you more specifically that we can recapitulate known motifs and peptide length distributions for monoallelic and multiallelic cell lines. I showed you that directDIA identifies more good binders than library-based analysis, and that the identified peptides are well quantified. And lastly, the unbiased directDIA in Spectronaut 20 is suitable for analysis of even highly variable and complex patient samples. With this, I would like to thank you for your attention, and I hope you enjoyed the presentation.